I'd like to first thank the moderator for pronouncing my last name correctly. It doesn't happen often. And I'd like to thank the organizers for inviting me to give this talk, which is basically the culmination of my graduate work in Christina Leslie's lab. It was a close, So this project was a highly collaborative project with Christine Meyer, where we were exploring the phenomenon of alternative cleavage and polyadenylation in human and trying to identify its pattern and consequence across the entire transcriptome. So just a brief introduction to the phenomenon that we'll be describing. So we know that Pol2 is recruited to the DNA in order to transcribe and activate a particular gene of interest. At some point, this polymerase will cross a finish line, which is defined by a number of sequence motifs that are transcribed into the RNA. And these sequence motifs are recognized by RNA binding proteins in the nucleus, which are recruited to the RNA. They cleave this actively transcribed transcript from the polymerase and then free it from Pol2 and allow it to mature into a functional mRNA. So we know that the 3' UTR is a host of a large number of elements that bind and regulate the mRNA post-transcriptionally, right? From microRNAs, which predominantly degrade transcripts or inhibit translation, to other RNA binding proteins, which may do the same or localize transcripts of different parts of the cell. Now, if you pick a gene out of a hat, you have a good chance of finding genes which have multiple functional finish lines, let's say. And when the cleavage machinery recognizes alternate polyadenylation signals, you can produce a full-length coding sequence of mRNA, which has differential 3' UTR length. And when we cleave a short form of the UTR, we free this transcript from the RNA binding elements that may affect the life cycle of this mRNA. So our interest in this project and a lot of other labs' interests as well may be summarized in this figure from Christine Meyer's paper in 2009. And what I'm showing you here is a northern blot of cyclin D1 in a large panel of cell lines. These are breast, these are lung. The light colors are normal tissues, normal epithelial cell lines. The dark are a transformed tumor line. And what we see is this top band is a full-length cyclin D1. This fast-migrating band at the bottom is the short form of the same gene, different only in the 3' UTR. And she found that there is an enrichment of expression of the short form of the UTR in cancer lines when compared to the normal. So not only does this happen at the RNA level, but she also showed that this has a functional impact on the protein output from a particular transcript. So using a luciferase reporter construct, either built with a short form of cyclin D2's UTR or a long form across a large number of cell lines, she showed that the short UTR form of the reporter can produce anywhere from 10x to 40x more output than its long form. So cleavage in differential UTR cleavage has a functional consequence to the amount of protein that an mRNA can produce. And so this is in the pathological context. Previous reports have shown in normal physiological contexts that there's large-scale differential UTR processing under normal physiological conditions. So just to summarize a few findings, it has been shown that in T-cell activation, we have a large shortening of 3' UTRs under a large number of transcripts. Likewise with synaptic activation. And it was shown in the mouse that during differentiation from ESLs to some type of neuronal cell, UTRs predominantly get longer. So we wanted to quantify APA, alternative polyadenylation, at this scale across the entire transcriptome in humans. So in order to do so, we developed our own sequencing library called 3'-seq. Now imagine these are RNAs from your lysate from the same transcript. This is a short form of UTR, this is a long form. So our protocol goes as follows. It starts with these oligo-DTs. This T-stretch is used to pull down the polyadenylated transcripts. This anchor encourages the oligo-DT to bind at the last templated base of the mRNA. And you see we have this one ribo-U in this T-stretch, which is kind of the secret sauce in our recipe. So what happens is the oligo-DT is used to prime reverse transcription. So we have this DNA-RNA hybrid. We use RNA-SH to digest the RNA, DNA-contacted nucleotides, and then we do second-strand synthesis to have a double-stranded DNA construct. Notice that we still have one. Notice that we still have one ribo-U here, and now we can use RNA-SH again, and it induces a nick here. By a process known as nick translation, we could push this nick upstream back to the transcript as far as we want. It's a time process. So we do that for about 50 nucleotides, at which point then, we could induce double-stranded breaks at the nick, allowing us to construct a library that only consists of the last 50 to 100 base pairs of the transcript in your sample. So now we have a sequencing library that's just the tails of our transcript. We take these reads, we process them, we align them to the genome, and now we get data that looks like this. So this is HN1 in a particular sample. We see a large increase of the expression of the short UTR as compared to the long form of the UTR. Now, if you look at this gene across different samples, so MCF10A is a normal breast epithelial line. This is a tumor breast line. And we see the same pattern that Christine noticed that she identified in her paper, where the short form of the UTR is preferentially expressed at a much higher rate than the long form in the MCF10A. So just to introduce some terminology, we just summarize shortening and lengthening by something that we call the short UTR index, or the SUI. It's simply just a fraction of reads that align to the proximal cleavage site. So in this example, the SUI is 0.9, in this example, the SUI is 0.5. Now, our data, I'm showing you a northern blot of the same transcript in the same, in the same cell lines, and our data accurately recapitulates the pattern of cleavage. So there are short and long isoforms here, as well as the relative expression that we've identified in our data. You see the same pattern in northern blots. Also note that the level of expression of these two genes is largely similar, right? So the only thing different between these two is the relative expression of the proximal or distal isoform. Okay, so we've actually run this protocol in a large variety of RNA samples. 7 of them come from primary tissues, and we have another handful from different cell lines. And there are 2 observations to make here. So one is, after what we think is a reasonable type of filtering, you can split the human transcriptome into 2 classes of genes. One of them is one class of genes only expresses a single UTR in any cellular context. The other will express 2 or more. The breakdown between these 2 different categories is about 51 to 49%. The other observation to make is, when a gene expresses multiple 3' UTRs, the position of the cleavage doesn't change between different samples. The only difference that we see is the relative abundance of the short versus long isoforms, so the regulation of APA in different samples. Lastly, I just want to point out that we could use R3'-seq as a proxy for gene expression. You could just add up the total counts in each of these peaks, and we can get an idea of the gene expression between these 2 samples. So we use both of these, gene expression and relative usage, to analyze APA in our lines, right? And so lastly, I want to point out, although I'll be talking about differences in APA using the SUI, this relative fraction at a proximal site, we actually identify differential usage of short versus long isoforms between conditions using this GLM, which was developed in this DEX-seq package, which is traditionally used to identify differential usage of exons between conditions, right? And so this is the formula for the GLM. It's really not important for this context. It's published and widely used already in the field. So the first set of examples I want to point out have to do with starting with cell lines that we've perturbed in some way, and we want to see how changes in expression and APA respond to these perturbations. So we'll start with naive B cells. We've transfected them with EBV and transformed them into these BLCL cell lines. What I'm showing you is a picture that everyone in this room looks at all the time, which is just a summary of differential expression and an MA plot between these 2 samples. So on the x-axis, we have the mean expression of a particular gene in these samples. On the y-axis, north to south, these genes are expressed higher in BLCL and these are lower, right? And we see, when we analyze the genes differentially expressed, we do your Go analysis, you find Go terms that you expect to be relevant in this experimental context. Now I'm going to change this picture slightly to something that we're not used to seeing yet. So x-axis still is the mean expression of the cell, of the gene in these two samples. Now north and south is just a difference in the SUI of the gene between these two samples. So as we go north, the UTRs get shorter in BLCL. As we go south, they get longer. And the interesting thing here is that the GO terms that are enriched when you look at differences in APA alone, ignoring gene expression, they largely recapitulate or reinforce the same transcriptional program that we've identified with differential expression. The perhaps more striking thing is when you combine the visualization of these two. So what I've done here is I've taken this plot, and the x-axis is now this north-south transposed. So as we go right to left, the gene is higher in BLCL versus B cells and lower. And what you see is the genes that are changing transcriptionally during this transformation are largely mutually exclusive from the genes that are changing their relative short versus long isoform usage. So it's as if APA is establishing a secondary axis of regulation, which we're blind to when we only look at differential expression, but the genes affected by both are reinforcing each other in terms of the functional program that these mRNAs are producing. Just to point out, we did the same exact experiment using MCF10A cell lines, and we have a constitutively active form of an HRAS expression, and we see the same pattern. So largely exclusive patterns of differential expression and APA regulation, but the functional terms that we get are the same. They reinforce each other. So this was an exploration of a particular pathway, activation of a pathway in cell lines. We want to look at what's happening in normal RNA tissue. So I said we have RNA from 7 different primary tissues. And then when you look at the distribution of genes that only express that only have one UTR, or genes that have multiple UTRs, in the subclass of genes that exhibit tissue-restricted expression. So I'm looking at genes that are only expressed in 1 or 2 tissues in the 7 that we have. You see that the large majority of these genes are single isoform genes, where 27% of them are multi-isoform genes. Now, if we expand, if we look larger, and now we focus on ubiquitously expressed genes, so genes that are expressed in 6 or 7 tissues, you see a very different pattern. So the majority of genes expressed ubiquitously have multiple 3' UTR isoforms, right? And so I want to further stratify genes by the level of APA regulation that we observe across our atlas. So we could do this, we can make 3 different categories. The 2 broad categories are genes that, so I'm focusing on multi-UTR genes. So there are 2 broad categories here. One of them is, there are genes that look largely like this. They express multiple UTR isoforms, but the relative abundance of the short versus long is somehow constant across the different tissue types. So there's a second class of genes, which we call polyadenylmodulated, right? So these are genes that exhibit some type of regulation at the 3' UTR level in some tissue type. So these type of genes are further stratified, and I'm mentioning this now because later I want to make a strong observation about these, right? So the genes that are like this, they exhibit, they're short in a number of cell lines, they're long in another number of cell lines. So here maybe half of them are short, half are long. When you look at these type of genes, which exhibit switch-like behavior in their APA, so in B cells here, this gene is not only short, but it's a lot shorter in B cells than they are anywhere else. And this gene here in brain is a lot longer in brain than it is anywhere else. So these are also PAM genes, but they exhibit the switch-like behavior. And we're going to look at how this plays out in terms of functional classes of genes in a minute. So the first thing I want to point out is that when you look at single isoform genes, NPAM genes, or PAM genes, so when we look at genes that have increasing levels of regulation or modulation by APA, these genes are both structurally and functionally distinct, right? So in terms of structure, the 3' UTRs get longer and longer with increased evidence of APA regulation. The genes that are multi-isoform and are in our PAM class, so they exhibit regulation by APA, whereas these don't, these genes have, on average, a proximal cleavage site that is a lot shorter or closer to the coding region than these multi-isoform genes, which don't appear to be regulated by APA. This just recapitulates the same thing. So the distribution, so the variability over the proximal cleavage site of the same gene across our 7 tissues is much larger in these PAM genes than NPAM genes. And lastly, the Go terms enriched for these 3 different classes are quite different. So these are somehow more housekeeping in functionality. Unique to these PAM genes, I have to do a lot with transcriptional regulation and specific types of signaling. Okay, so lastly, we've seen tissue-restricted versus ubiquitous and how single isoform or multi-isoform genes are stratified between the two. But if you look at increasing expression breadth, we know that the number of multi-UTR genes also increases, but moreover, more than just that, the proportion of genes that are regulated by APA is increasingly increasing with increasing breadth of expression. So as genes are expressed in more cells, they have a higher tendency to be regulated by APA. So I want to actually focus in this group of genes, so these PAM genes, specifically the ones that exhibit the switch-like behavior that I mentioned to you before. So what I'm plotting here is, this is a heat map, which is just the SUI, right? So it's the length of the UTR in a particular sample. High red means that the UTR is extremely short. Deep blue, the UTR is extremely long relative to each other, so to speak. And so what we see here is first, there are some tissue types that exhibit a rich APA landscape. So in testis, there's a large number of genes that are, I want to stress again that these are ubiquitously expressed. They're expressed in all 7 cell tissue types. So there are a large number of genes that are extremely short in testis when compared to the UTR relative expression in other genes. B cells have a very interesting mix of short and long 3' UTRs when compared to everything else. Not much seems to be happening here, and brain exhibits the longest UTRs in our tissue atlas, and this phenomenon has been widely reported already, so this isn't anything quite new. But the interesting thing here is when you do Go analyses on these exclusively short, long genes, ignoring differences in gene expression between the 2 tissues, we recover tissue-specific functionality that's relevant to the tissue type that we're looking at, right? And that's summarized in this Go analysis here. Furthermore, as we look at UTRs of genes that are increasingly modulated by APA, we find a very interesting pattern of conservation around the regions that are bound to and recognized by the cleavage machinery that induces the cleavage. So this is a pseudo UTR, which is summarized over the, all of the multi-isoform, all of the multi-3' UTR genes we have in our atlas. This red line identifies the cleavage site, is the cleavage site for the particular gene. That's proximal, and here's the distal, so cleavage site for these genes. And this part here is just random sequence somewhere between the proximal and distal. And so what you see here is multi-isoform 3' UTRs that aren't regulated, that aren't our PAM set of genes. You see an increase of regulation around the regions that are recognized by the cleavage machinery. If we then look at multi-isoform PAM genes, so these are specifically short or long in some number of cell types, not these switch-like behavior, you see an ever-increasing amount of conservation around the regions that are important for the machinery to recognize to cleave at this site. And lastly, the genes that exhibit this tissue-specific switch-like behavior are even further increased. They have a further increase of conservation around these sites, suggesting that these are quite, I mean, you know, we're using a proxy of, conservation here as a proxy for functional requirements of the sequence around the RNA there. And the last little vignette that I want to point out is an interaction. So we were exploring different types of interactions between APA and other post-transcriptional regulatory mechanisms. And the most widely, I guess, studied post-transcriptional regulation mechanism are microRNAs. And so this is somehow a complicated figure, but the summary, the punchline can be given by this figure here. So what I'm showing you here, these are the single NPAM and PAM genes. So these are regulated by APA. This region in this blue site is the median proximal cleavage site, and this is the part that is regulated by APA and can be preferentially kept or lost. This part here is a constant region. APA can't do anything to include or remove this. It's always there, just like the single UTRs. And what we saw is that when you compare binding of microRNAs to variable regions of UTR versus constant ones, we found an enrichment of binding of ubiquitously expressed microRNAs to ubiquitously expressed genes in the variable region when you compare binding to here versus here. So I'm showing this result in this waterfall dot plot here. So each one of these points is a p-value from the test of enrichment of a microRNA binding to the distal versus constant region. And what we see is we tested about 87 microRNAs for this, and we found, this is negative log 10 p-value, and we have an ever increasing number. And with the lower p-value, you see it scaling up the graph here. This gray curve in the background is the same plot, but here we've generated just random microRNA seeds, and we're calculating the same statistic, enrichment of binding in a variable versus constant region. And so this is serving as our empirical null distribution, right? So we are getting lift off of our empirical null, and the dots, so in ubiquitously expressed genes that are regulated by APA, the dots that are largely above background are these green dots, which are ubiquitously expressed. If we do the same test for the NPAM genes, so multi-isoform genes that don't exhibit modulation by APA, we don't see any difference from background. And lastly, if you look at microRNAs that bind single UTRs versus the preference to bind single versus regulated regions of UTRs, we find that the microRNAs that are enriched to bind here versus here tend to be ones that are tissue restricted. They have a tissue restricted expression pattern. So that kind of concludes our advertisement for this paper, which was published in Genes in Development at the end of last year in 2013. And so just as a recap, one of the main points of observations that we're making is that APA is an additional modality of regulation that preferentially targets ubiquitously expressed genes in a tissue-specific manner. I've shown this in several ways, I think. So here, this is the secondary axis of regulation by APA versus differential expression. They're largely mutually exclusive. As genes increase in their breadth of expression, we see increased incidence of regulation or modulation by APA. These genes that are expressed ubiquitously in all 7 tissues that exhibit switch-like behavior are enriched for Go functionality that is specific to the tissue where the switch-like behavior is observed. And lastly, this conservation pattern suggests that the regulation of APA around these sites is increasingly important, from switch-like to regulated, non-regulated, and this preference for ubiquitous microRNAs to target variable regions of ubiquitously expressed 3' UTRs is the last point. So with that, I'd like to thank Christina Leslie, who is my graduate advisor, runs a great lab and is the type of scientist all of us can aspire to be. She's looking for talented computational postdocs. So if you're looking to do a machine learning heavy type of work or more collaborative biology, more biologically motivated work, you can find both of them there. And we know that there's great research happening at Sloan Kettering. Julie Yang was a junior graduate student in the lab who performed the microRNA analysis. I'd like to thank, just very briefly, the members of Christina Leslie's lab who had a large overlap with my graduate career. In particular, Anshul Kandaji was a senior graduate student when I was just a rookie graduate student, and I learned a lot from him, and I could ask him questions that I was too embarrassed to ask Christina in the beginning, right? And so Christine Meyer was the biological force behind this analysis, And Utr Garg is a research technician in her lab who helped with some of the experiments that I showed you today. Obviously, Bioconductor and core community for providing all the tools that we use to analyze all of our data, and some funding resources. And with that, I'm happy to take any questions if you have them.
Thank you. That was a great talk. It was interesting. You started
Can you
off.
present yourself?
Yes, Menachem Fromer, Mount Sinai School of Medicine in New York.
Nice.
Yeah,
Okay.
So, so you started off with the motivation looking, or you mentioned that when you have these different poly tracks, then it actually results in differential expression at the protein level. Did you follow up on any of those in particular for particular examples of interest, or thinking of doing a proteomic experiment to validate that really this does ultimately leads to differential translation levels?
We did think about doing it.
Okay.
We did.
Yeah.
So we don't have examples at this scale. I mean, not included in our paper or not published. Christine has several examples where we actually exhibit both, right? So different UTRs will sometimes. So the naive assumption, the first approximation, you would think a short UTR will express more proteins. We definitely see that, not just with the reporter construct, but we also see the reverse, which is also, I'm not sure what that means, but that's what it is. But we don't have any validation at this scale at the proteomics level. We'd like to, but I think that's quite a, well.
Cool. So you left work for other
Exactly. Yeah. I
Okay.
mean, we can't do everything, right? So.
Thank you.
Thanks.
Hi, I'm Kevin Huff from University of Toronto. Thanks for your talk. I was just wondering if you have any insight on what's going on with that little peak in conservation just upstream of the cleavage site.
Yeah, that's so. You mean
Yeah.
right here?
Yes.
So we know that, so about from 10 to 40 base pairs upstream from the cleavage site is the actual polyadenylation signal. And that's the main motif that is recognized by CPSF. There are other kind of motifs that are uncharacterized that are around the polyadenylation signal, both upstream and downstream, that are relevant for increased or decreased recruitment. But this large peak here is definitely a result of the polyadenylation signal, which is 10 to 40 base pairs upstream from the cleavage site.
Thanks.
Yep.
I'm Julie Zhu from UMass local medical school. And so we did some PASIC. I don't know if you're aware of that.
Yeah, definitely.
So, yeah. So what's the difference between your protocol and the PASIC protocol? And then the reason I ask is PASIC has this internally primed signal, like a sequence.
Yeah.
So it's like it's false positive, false
Yes,
IUDR.
yeah, yeah.
I'm just wondering, your protocol has that problem too?
Oh, we definitely do. But so I mean,
how do you deal with that?
yeah, so I didn't have time to go into it here, but we actually have a pretty extensive pipeline, which we actually hope to release to Bioconductor for its next release, that identifies internal stretches of long A stretches internal that seem to be false positives, and we remove them with some heuristics in terms of the poly A stretch, if there's a polyadenylation signal close by, or other things. But I mean, it's in our supplemental, I think we went into details of that. I'm happy to chat with you about it more if.
Yeah, because we published a paper in bioinformatics. Actually, it's Bioconductor package.
Oh, yeah?
Yeah, to remove that, use the naive Bayes approach. Just wondering if you
Yeah.
like
So-
compare the.
Yeah, I'd love to talk to you about that,
Okay.
actually. That'll be interesting. Thank you.
Can I ask
Yeah,
a simple
definitely.
question? You mentioned, and it makes sense, that a lot of dysregulation maybe is somewhat are reflected through the microRNA, as you show us in there. So if you do the same analysis or similar analysis, let's say for mice or for, yeast that have no microRNA, would you get something that will reflect it? And I mean, I'm trying to get the microRNA component out of the story.
That's a great question. So we haven't looked at mouse microRNA binding or yeast APA. But I mean, the only thing I could say is that the hits that we consider for microRNA binding they have to be conserved sequence across, I think it's 5 out of 6 different animals, one of which is mouse and rat, right? So but we didn't look at patterns of microRNA expression in mouse, whether they're tissue restricted or ubiquitous, and we didn't do this analysis in mouse. But Christina showed me a very interesting result in a paper that they're under review, which I think challenges a lot of what we think we know about microRNA regulation. So there's still a lot that we don't know. Yeah.
Please.
Yeah.
Maybe last, yeah. Okay, go ahead.
I'm Tatiana Tatarina, USC. In my lab, we are studying the effect of variability of 5' UTR onto gene expression.
Cool, nice.
And my postdoc actually having a poster about it. So if you
Today?
check it out, yes.
Oh, awesome, nice.
And but what I was wondering, there seems to be lots of similarities between 5' UTR effect on expression and 3' UTR. Have you considered looking at them at the same time?
Yeah. So if I had my way, we would. So there are some sequencing protocols where you could circularize the transcript, and then you could tag the 5' and 3' end and pull them down together, so you know what 5' end goes with which 3' end. And my dream would be to have such data to analyze that. We do know that. So we do know that transcription factors can assist in determining poly-A signal choice. Right. So Christine actually has some interesting work that's not published that shows messing with TF binding can change the polyadenylation signal choice at the end.
That's fascinating.
Yeah, it's really crazy.
Thank
Yeah.
you.